Okay, well, welcome to our afternoon Safe World by Design webinar today, and I'm very happy to have Manuela Pavan with us from Innovatune, and we've had some good interactions over the years, and she's going to concentrate on today, on how in silico methods can actually be useful in terms of practical risk assessment. So it's hopefully an important topic for your work. The, the lecture will probably take something like 20-25 minutes. You can enter a question in the chat or question box at any time, or we can just open it up after Manuela gives her presentation. So looking forward to it. I'll go away and join the audience now, Manuela, and off you go.
Thank you. Thank you very for this introduction and good afternoon, everyone. So welcome to this webinar where I will focus on the topic on how can in silico methods support the derivation of safety limits. Let me just introduce myself. I'm Manuela Pavan from Innovatune, a consulting company making the most of in silico methods. I'm a computational toxicologist providing consulting services for evaluation of human health endpoints. And I founded Innovatune together with my colleague Ariana Bassan some years ago. And here in Innovatune, we provide consultancy services in toxicology in different contexts, mainly pharmaceuticals, but also agrochemicals, medical devices, cosmetics, and industrial chemicals. In the webinar today, I'm going to show you how in silico methods can be used to support the derivation of safety limits in different regulatory contexts, like, for example, the context of genotoxic impurities, non-genotoxic impurities, occupational bending, and extractable and leachables. As a background, and also just to share a common terminology, let me say that the term in silico methods refers to approaches that use the chemical structure of a molecule to predict its toxicity. So the starting point, the input information in the so-called in silico methods is the information coming from the structure of a given chemical. In silico methods include the so-called QSAR methodologies, where QSAR stands for quantitative structure activity relationships. QSAR models are mathematical and computer-based theoretical model that can be used to predict in a qualitative or in quantitative way the activity of compounds, starting, as we said, from the knowledge of a chemical structure. QSAR models can be statistically based models or expert rule-based models, and these two different methodologies have different advantages and disadvantages, and as a consequence of that, their combined use is often suggested and recommended in many different regulatory and scientific contexts, and I will come back on this concept in a few slides. Computational methods can support the chemical safety assessment in a convenient and in a knowledge-based manner. However, the integration of in silico predictions in regulatory frameworks to define quantitative safety limits is still not much explored. This might be to the fact that the in silico methods are approaches that can robustly support the hazard identification, while they are less able to support hazard characterization. In fact, in silico methods are mainly, or in most cases providing predictions in terms of yes or no, positive or negative, thus in terms of qualitative predictions rather than quantitative predictions. The aim of this webinar is to present possible ways of the use of in silico methods to support the derivation of quantitative limits in specific regulatory context for data-proof substances. And in more details, two approaches will be presented and discussed here today. The first one is the so-called class assignment approach. This approach applies in those contexts where classes and corresponding limits are already established by toxicologists, meaning that a quantitative limit is already associated to a given class by default. The definition of the classes and corresponding limits depends on the specific regulatory context or by the specific application field. So in this, and according to this approach, in silico methods can be used to predict the endpoint of interest. And based on the prediction that is provided by in silico methods, usually the yes or no, the target molecule is then assigned to a given class and corresponding limit. One of the most well-established use of in silico methods for regulatory purposes, according to this type of approach, so according to a class assignment approach, is represented by the ICHM-7 guideline for genotoxic impurity. The ICHM-7 guideline is the guideline for the assessment and the control of DNA reactive mutagenic impurities in pharmaceuticals. And the hazard assessment process of the ICHM-7 is based on the assignment of any impurity to a given class with associated limit. The workflow, the hazard assessment workflow that is implemented in the guideline is described in the slide that you can see now on my screen, so in the picture that is presented here. According to this approach, the hazard assessment is firstly performed by searching and by collecting potentially available experimental data of bacterial mutagenicity and carcinogenicity. And if good data are identified, are retrieved, then these data are used to assign a given impurity to class 1, which is made of impurities that are mutagenic and carcinogenic, or to class 2, that is made of impurities that are known to be mutagenic but with an unknown carcinogenic potential, or to class 5 that is the class composed by impurities that are assessed as not mutagenic. However, if data are not available for bacterial mutagenicity and carcinogenicity, a QSAR analysis can be conducted, and based on this QSAR analysis, impurities can be assigned to class 3 if the prediction resulted to be positive. In class 5, if the prediction results to be negative, or in class 4 that can be considered negative despite the identification of a structural alert based on the fact that the same alert is identified in a similar chemical environment in an experimentally tested negative compound that in most cases is the IPI itself or a related impurity. The ICHM-7 guideline specifically defines how in silico methods and how the computational toxicology assessment can be performed in this context. In fact, it's mentioned in the guideline that a computational toxicology assessment should be performed by using QSAR methodologies that predict bacterial mutagenicity assay. So the endpoint that is used for this class assignment is bacterial mutagenicity. The guideline then also states that the two QSAR methodologies that complement each other should be applied. One methodology should be an expert-based rule system, and the other one should be a statistical-based approach. And the QSAR models that are used to derive the predictions should follow the OECD principles for QSAR validation, meaning that the models that are used for this class assignment, for this hazard assessment, need to be compliant with regulatory requirements. According to the guideline, the absence of a structural alert from the two complementary QSAR methodologies is sufficient to conclude that the impurity is of no concern, and in such a case, no further testing is recommended. I would like also to emphasize here that this guideline also highlights the importance and the role of an expert review. In fact, the guideline states that the outcome of any QSAR system-based analysis can be reviewed by the use of expert knowledge. And this is needed and is always recommended in order to support evidence on any positive or negative prediction. But it becomes even more crucial when we are dealing with conflicting results or inconclusive prediction, where in such a case, an expert review is needed in order to provide the rationale that supports the final conclusion. The ICHM-7 guideline indeed represents a milestone for the acceptance of in silico methods for the hazard assessment in the context of pharmaceutical impurities. But this guideline has also influenced and promoted the use of in silico methods in other contexts, for example, in the context of veterinary medicinal products, in the context of extractable and leachables, where the mutagenicity assessment is performed according to the ICHM-7 guideline, and I will come back on this specific application field, but also in the context of genotoxicity assessment of pesticides in more details with respect to the bacterial mutagenicity assessment according to the approach that is suggested by the European Food Safety Authority. A class assignment approach is also adopted in the context of an occupational exposure banding. An occupational exposure band is defined as the range of airborne concentration levels expected to be protective of the worker's health. And we have to keep in mind that each band is then associated to an upper and a lower occupational exposure limit. In this specific context, chemicals are assigned to a given band and therefore to the corresponding range of occupational exposure limit they used based on the toxicity of a chemical. And the assessment of a toxicity might also make use of specific JHA's category. In this specific context, in silico methods may complement the toxicological profile of data pool substances. So it meaning that in silico methods can be used in this specific context to provide toxicological information, but also JHA's categorization for those chemicals that are lacking experimental information on these elements, on the toxicological profile. Mutagenicity, also in this context, is always conducted according to and in compliance with the principles that are defined in the ICHM-7 guideline, meaning that always employing two different methodologies that complement each other, the statistical-based and the expert-based system. In the next step, if no concern is identified for mutagenicity, the endpoint-specific criteria of the underlying schema, on the underlying bending schema, can then be used to assign the target chemical to a given category. In this specific context, there is no unique or harmonized approach for bending categorization, meaning that there are, in the literature, there are available and have been proposed many different bending schemes that can categorize chemicals based on the experimental or predicted toxicity. And the table that is represented here is extracted from a poster that we presented this year. It was in last March at the SOT Congress. And in this table here, you can see how, based on the specific profile of a chemical, a corresponding band is identified. So what we commented before is that of course, this toxicity profile might be based on experimental evidence when this evidence is available. However, if the experimental evidence is not available, so when we are dealing with data pool substances, the toxicological profile can be assessed by means of in silico methods. And based on the toxicological profile that is, in such a case, predicted, a given band can be assigned with then the associated upper and lower limits related to that given band. As mentioned at the beginning, in silico methods can also support the derivation of quantitative limits through another type of approach, that is the so-called read-across approach. The read-across approach is one of the most widely used methods to fill in data gap in human risk assessment. And in the read-across approach, the information of a given property or of a given endpoint for one or more sources that are the so-called data-rich chemicals is used to derive the information to predict the property of the endpoint for one or more target chemicals that are lacking this experimental evidence, so the so-called data pool chemicals. The read-across approach is based on two key elements. The first one is the hypothesis of approach and the justification of approach. The hypothesis of a read-across approach is that the property, the endpoint of the target chemical can be inferred from the available data of one or more source compounds. And this indeed involves the identification of similar suitable analogues. The second element consists in the read-across justification, which is aimed to provide the rationale to support the use of available experimental data of the analogues to predict the property of the endpoint of the target chemical. And this justification is focused on similarity considerations of the analogues as compared to the target chemical, where these similarity considerations really are crucial and they drive the read-across argument. In the read-across approach, indeed, the similarity is the key concept, and it requires the comparison of the analogues with respect to the target according to different perspectives. In most cases, the first perspective is the structure similarity one. So it means that the analogues are compared with respect to the target in terms of their structure similarity. However, this similarity analysis needs to take into account also other perspectives. For example, a physico-chemical similarity, but also an ADME, a metabolic similarity, and also a toxicological similarity. And any difference, any relevant difference that is detected in this similarity analysis does not necessarily discard a given analog, but it has to be addressed and commented carefully. For example, it can support the selection of a worst case scenario. In fact, the differences in this approach are justified whenever the source presents additional elements of concern with respect to the target and not vice versa. So meaning that we want, of course, the source to be sufficiently conservative as compared to the target. In this slide, you can see how structure similarity and the physico-chemical similarity can be performed. On the top of this slide, you can see that we have a table here where we compare the structure of the target with respect to the one of the analog. This structure similarity can be performed by means of similarity index, for example, by employing Tanimoto similarity using specific fingerprints, but it can also be performed by employing specific profilers, such as those reaction domains that are designed in order to identify critical fragments. On the bottom side of this slide, you can see the physico-chemical similarity table that is presented here. You can see that in this, when we assess the similarity of the target as compared to the analog in terms of physico-chemical properties, we can fill in this table where a number of physico-chemical properties are presented and provided in the table for the target and for the analog. The physicochemical properties that are provided here can be, of course, based on experimental evidence whenever this is available, or again, could be predicted data. And the physicochemical properties that are relevant, that need to be taken into account in this physicochemical similarity really depend on the specific endpoint that we are processing, depend on the specific endpoint that we want to read across. However, generally speaking, we can say that the molecular weight, for example, water solubility and lipophilicity, log-KOV, are physicochemical properties that are always taken into account when we compare an analog with respect to the target. Another similarity that often plays a crucial role in the context of a read-across approach, and that I could also say is one of the most challenging, often most challenging element of a read-across study, is the metabolic similarity. In fact, the metabolic similarity is relevant in the context of read-across, since it's not only the intrinsic biological activity that is causing a given effect that is important, but also the ADME profile that at the end defines the internal exposure of a given chemical. Metabolic similarity can, of course, be addressed by means of literature review, by collecting evidence and information when this is documented in the literature, but it can also be supported by in silico methods. For example, by using ad hoc profilers, like, for example, the bioactivation profiler that is implemented in the LeadScope model applier, that is designed in order to identify potential structure features which may result in the formation of reactive metabolites. According to this approach, again, you can see the table that is displayed on the top of this slide. We can compare the target and the analog by means of general bioactivation profilers in order to compare the reactive site of metabolites that are identified in the target and in the analog. Another approach, of course, another method that can also support a metabolic similarity is the rat liver metabolism simulator that is implemented in the ACD-QSAR toolbox, which allows, for example, comparison of metabolites coming from the target with those that are coming from a given analog. And of course, a following step would be also to evaluate and to assess the toxicological behavior of the metabolites as coming from the target with respect and as compared to those coming from the analog. Finally, but not for this reason less important, is the assessment of toxicological similarity of the analog as compared to the target. And this is another very relevant and crucial piece of information in the context of read-across justification. Since the target, as we commented before, is a data pool chemical, the analysis of the biological similarity of the toxicological similarity of the analog with respect to the one of the target is often performed by comparing the predicted toxicity profile rather than the experimental one. So it means that the prediction of the toxicity profile is used in this case to justify the suitability of a given analog in the context of a specific read-across. In this table here, you can see how predictions for different endpoints are displayed for the target compound and for the analog and are compared each other. The endpoints that are included in this toxicological similarity, again, depend on the specific read-across use case. So again, depend on the specific endpoint that we wanted to read across. Indeed, a broad assessment of toxicological similarity, keeping into account a broad range of endpoints, is suggested and recommended. Since we are dealing in this case with predictions, an expert review, of course, is always recommended and suggested for the analysis of this toxicological similarity. Let me also emphasize that with respect to this toxicological similarity, the goal here is really to determine whether the target has a similar biological behavior as compared to the selected analog, meaning that what we do not want is that the target trigger adverse effects that are not covered by the analog. And this is the reason why the expert review focus on confirming or dismissing any difference that may arise from this specific analysis. So the approach here is not aimed to predict the actual toxicity of the chemicals of the target and the analog, but rather on comparing the toxicological behavior of the analog with respect to the one of the target. Once the read-across approach is justified, the point of departure for the target can be derived from the critical study, for example, the repeated dose study data that are available for the analog. And the point of departure here could be used to establish a safety limit for the target. And of course, in that case, uncertainty factor could also be applied in order to take into account that a read-across approach was applied. A context where in silico methods are increasingly used to support the derivation of safety limit is the one of extractable and leachables. Extractable and leachables are organic and inorganic compounds that originate from the components that are used in the packaging, and these compounds have the potential to adversely affect the safety of the drug products. It's important also to highlight here that extractable and leachables impurities are currently excluded from the scope of general ICH guidelines. And this really is a field where there are ongoing activities that are carried on with the main purpose of better defining how QSAR models can be used to support the qualification of extractable and leachables. The workflow that is currently followed in particular for what concern parenteral drugs is the one that is documented in the PQRI document dated back in 2021, so in the Product Quality Research Institute document. And according to this approach, the workflow here aims first at assessing the mutagenic potential according to the workflow that is proposed in the ICHM7 guideline, so according to the ICHM7 approach. So again, employing two different QSAR methodologies that complement each other, a statistical-based approach and an expert rule-based approach. And for an extractable and leachable that is assessed as positive based on experimental evidence or based on in silico evaluation, then the limit here and the qualification is set to 1.5 microgram per day, the TTC limit. In the next step, a weight of evidence approach is envisaged with respect to irritation and sensitization, which also takes into account the use of in silico approaches. And at this stage, if an extractable leachables is identified as a strong irritant or sensitizer, the qualification limit is set to 5. So the next limit, let's say after the 1.5 microgram per day, is set to 5 microgram per day. However, higher limit can also be derived from the toxicity assessment based on other endpoints using a read-across approach. And let me also say that this we know that is the current status, but we also know that there is activities that are ongoing and that are expected to become available with the upcoming ICH Q3E guidance, where these limits may also be revised and where further details on how to employ in silico methods for the safety limited derivation of extractable and leachables will be documented and defined in more details. The two approaches that we commented today, the class assignment and the read-across approach, are not necessarily used in, they can often be used in a complementary way. So it means that in many and different contexts, these two approaches are used together. This is the case, for example, of limited derivation for genotoxic and non-genotoxic impurities, where in the first step, class assignment approach is applied in order to assess the genotoxic potential of a given impurity according to the ICHM7 approach, the ICHM7 guideline. And if the impurity is assessed as a non-genotoxic, in the next step, a read-across approach can be applied in order to support the derivation of a PDE according to the ICH Q3C and Q3D guideline. So in such a case, once established that the impurity is not genotoxic, in the case of a data poor substance, so again, in the case where we are lacking experimental evidence, a read-across approach can be used and can be employed in order to support a PDE derivation. So in this webinar, I wanted to present you how in silico methods can support the derivation of quantitative safety limits for data poor substances in different regulatory contexts. And what I presented here today are these two sequential approaches that can integrate the use of in silico methods for limits derivation. One is, as we commented, the class assignment, where basically in silico methods can be used in order to assign a given target compound to a class, which is then regulated and with limits that are defined based on the specific regulatory framework or based on the specific application field. And we saw that this, for example, applies in the context of ICHM7 guideline, but is also the case in the occupational banding, where we have a number of bands and where in silico predictions can support the categorization, the class assignment of a target compound. Another approach is the read-across approach, where in that case, the justification of the similarity of the analog as compared to the target really plays a crucial role. But once the read-across is justified, then the point of departure of the analog can be used in order to derive a quantitative safety limit for the target. Of course, taking into account of all the limits and uncertainty factors related to the read-across approach that was adopted. And the flexibility of in silico methods warrants their possible use in several application areas. We presented and we talked today of genotoxic and non-genotoxic impurities, but also occupational exposure banding and extractable and leachables. And with this, I would like to acknowledge my colleagues, Arianna Bassan and Christian Novello. And of course, I also thank all of you for your interest in this webinar and for your participation.